Welcome back to The Explainer. Today, we're zeroing in on an absolute microscopic titan. I'm talking about the molecule that literally powers virtually every single second of human life, ATP. Let's just dive right into the mechanics of this thing because I gotta tell you, the reality of how it actually works is way more fascinating than those basic diagrams you probably remember from high school biology.
I mean, have you ever actually stopped to think about how your cells pay for a heartbeat? When your heart thumps in your chest, or when you blink, or just take a breath? None of that happens for free. It all requires funding. Down at the cellular level, physical actions literally have a cost. We're talking every single heartbeat, every tiny nerve impulse firing in your brain right now as you listen to this, every muscle movement, whether you're lifting a heavy weight or just cracking a slight smile. None of it is free. They all require a very specific type of biological payment to function. And without this payment, everything just stops. So what exactly is this tender that your body accepts?
Well, here's our roadmap for the explainer today to figure that out. We're going to start with the cell's energy currency, break down the actual anatomy of ATP, bust what we call the broken bond myth, look at how this funds biological activities, and then wrap up with the next big question.
So, let's jump right into it with Section 1, the cell's energy currency. Let's see how life actually pays the bills.
Meet ATP, or adenosine triphosphates. This is the primary energy-carrying molecule found in literally all living cells. Almost every single energy-requiring process in your body depends, directly or indirectly, on this exact molecule. It acts as a sort of middleman, transferring usable energy from reactions that produce it to reactions that consume it. Bottom line, without ATP, life simply cannot be sustained.
Honestly, it helps to think of ATP exactly like human money. Just as we exchange cash for goods and services out in the real-world economy, your cells use ATP to exchange chemical energy. See, your cells extract energy from nutrients, like the glucose in that sandwich you ate for lunch, and they convert a chunk of that energy into ATP. So whenever a biological process needs to buy a little energy to get a job done, ATP steps right up to the counter and hands it over.
All right, moving into Section 2, the anatomy of ATP. Let's put this currency under the microscope.
So ATP is a nucleotide, and it's made up of three main components. First up, we've got adenine, which is a nitrogenous purine base. Second, we add in ribose, a 5-carbon sugar. Those two lock together to form adenosine. And then finally, we attach this tail made of 3 phosphate groups, the alpha, the beta, and the terminal gamma phosphate.
Now pay attention to how these pieces actually connect. A phosphoester bond connects our ribose sugar to that first alpha phosphate. But the really absolutely crucial connections here are the phosphoanhydride bonds. These are the ones connecting the alpha to the beta and the beta to the gamma phosphate. These specific phosphoanhydride bonds are the exact locations associated with ATP's whole energy-releasing transaction. That is where the magic happens.
To really appreciate what we're dealing with here, just take a look at the sheer chemical formula: 10 carbon, 16 hydrogen, 5 nitrogen, 13 oxygen, and 3 phosphorus atoms. It is a beautiful, highly complex piece of machinery, and it's operating trillions of times a second inside you right this very moment. Crazy, right?
Which brings us to Section 3, the broken bond myth. Let's talk about how this transaction actually works. Okay. So we're often taught this myth in school that energy is just neatly stored inside the ATP bonds, like money locked inside a little vault, and it just spills out when the bond is broken. Actually, scratch that. That's a massive oversimplification. The reality is so much cooler. Energy is released because when ATP is broken down, or hydrolyzed, the resulting products are just far more thermodynamically stable than the original ATP molecule was.
So why exactly are the products more stable? Well, first off, you get a relief of electrostatic repulsion. Those 3 phosphate groups are negatively charged and they're packed super closely together, basically repelling each other like totally compressed springs. Breaking them apart relieves all that built-up tension. Second, the resulting inorganic phosphate has greater resonance stabilization. Third, the products are just better hydrated by water molecules floating around in the cell. And finally, breaking one molecule into 2 causes a net increase in entropy, which, as we know, the universe absolutely loves. All these factors combined are what make ATP hydrolysis so incredibly energetically favorable.
Down on a purely chemical level, the transaction looks exactly like this.
ATP reacts with water, H2O, spending that terminal gamma phosphate we were talking about earlier. It pops right off, leaving behind ADP. That's adenosine diphosphate, di because it now only has two phosphates, plus that lone, highly stable inorganic phosphate. That thermodynamic stability we just broke down results in a standard Gibbs free energy change of approximately negative 30.5 kilojoules per mole. And that negative sign? That's the crucial part. It means energy is actively being released for the cell to use. And what's really amazing is that inside living, breathing cells, this number is often even more negative and powerful, simply because the natural concentrations of ATP and ADP in your body differ so much from standard lab conditions.
All right, moving on to Section 4, funding biological activities. Let's open up the cell's budget. So ATP funds a massive variety of biological work. For mechanical work, every single time your muscles contract or motor proteins move around inside your cells, ATP is footing the bill. Then you've got transport work, actively pumping sodium, potassium, and calcium across cell membranes against their natural gradients. That requires constant, non-stop ATP expenditure. And what about chemical work? Synthesizing your DNA, your RNA, proteins, lipids, all of it costs ATP. Finally, regulatory functions. ATP is heavily involved in cell signaling and regulating enzymes. It literally touches every single corner of your biology.
But here we run into another major myth. People think that your cells store massive vaults of ATP, hoarding it away like a dragon hoards gold. The reality is actually the complete opposite. Cells store only a tiny, tiny amount of ATP. Every heartbeat, every nerve impulse drains that supply incredibly rapidly. So rather than sitting around in large stockpiles, ATP is constantly and rapidly recycled. It's produced, consumed, and produced again in this ceaseless, furious loop.
Which sets us up perfectly for Section 5, the next big question, the biological mint.
This brings us to a pretty massive core mystery. See, ATP itself is not the ultimate source of energy. It's just the currency. It just transfers energy from one place to another. But currency has to be minted somewhere, right?
So the crucial point we are left with is this. How does a cell continuously produce ATP? If we only store a tiny little amount, yet we require literally millions of ATP molecules every single second just to survive, where is our endless supply of recycled ATP actually originating from?
Well, the answer lies in the fact that every living cell runs its own microscopic, unbelievably complex manufacturing factory. To mint our cellular currency, the body relies on incredible processes. Things like glycolysis, the citric acid cycle, the electron transport chain, and this absolutely wild microscopic rotary motor called ATP synthase, all driven by something called chemiosmosis. These are the systems that take the food you eat and spin it into the literal currency of life. But think about it. If a single cell requires millions of these microscopic transactions every second just to keep you alive, how does this microscopic mint keep up the pace without just entirely melting down? Well, we're going to dive into that amazing complex factory in our next explainer. See you then.